Well, first of all, to introduce a little bit the background of my study, MBL is, I work in low count MBL, which is a condition in healthy individuals that present very low numbers of clonal B cells in the peripheral blood. It is unclear if it represents a pre-leukemic condition or not, but what is clear is that it's very strong age-related. People which are older present more incidence of low-count MBL. Another topic of my research is clonal hematopoiesis, which is defined as the presence of a somatic mutation in a hematopoietic stem cell that provides this cell a better survival and a better capacity of proliferation, or a better proliferative capacity. The goal of our study is to determine the incidence of clonal hematopoiesis-related mutations in subjects which present a low-count MBL. We have conducted this study using 56 healthy donors and 106 subjects with low-count MBL. We studied in neutrophils, monocytes, and CD4 T cells a custom panel of 239 genes related to clonal hematopoiesis, also clonal cytopenia of undetermined significance, and myelodysplastic syndromes. After the sequencing using a NovaSeq X technology from Illumina and the bioinformatic analysis, we found that in these two groups, the healthy donors without MBL and the MBL cohort, we had a global similar incidence of clonal hematopoiesis-related mutations, around 50%, but which was surprising for us is that in the healthy donors, we could see a strong difference in age between the people which had clonal hematopoiesis-related mutations and the people which had not these clonal hematopoiesis-related mutations, being the mutated ones older. This is logical because clonal hematopoiesis is a strongly age-related condition. In the people with MBL low, we couldn't differentiate this. We couldn't see this age difference between MBL low with clonal hematopoiesis-related mutations and people with MBL low with no mutations. This could imply that it's not the age the main factor which is driven the appearance of these clonal hematopoiesis-related mutations, but a previous defect in the hematopoiesis that may lead to the appearance of these mutations, but also to the appearance of these B-cell clones characteristic of low-count MBL. Another finding which is interesting, or which was interesting, is that the mutational profile between the healthy donors without MBL and the mutated MBL was different. There was no mutations which were shared between the people with MBL and the healthy donors. This also is interesting. And we keep working in this project, and in the future we want to investigate, we are already doing it, we want to investigate the incidence of clonal hematopoiesis-related mutations also in subjects which present small numbers of T clones, which is known as TCAS. So we can have a broad spectrum or the whole landscape of the small clones that may appear in the peripheral blood. B-cell clones, T-cell clones, and also myeloid cell clones. With all this data, we want to see if the appearance of these small clones in peripheral blood may be driven not by age, not by environmental factor that surely play a role, but maybe it's driven mainly by a defect in the hematopoiesis, and the appearance of these clones is like a physiological response to compensate this defect in the hematopoiesis.